Dr. Londrich here. So I put together all your files and put together all the data that I had from the samples you sent, and I'm going to send those files to you. So one of the files is called Biology 106 Animals, and the other one is Biology 106 Fish DNA Sequencing Analysis. So this is one of your submissions. Pretty much all of them are the same, but I like this one in part because I had, um, well, it was the first one, and I also had the percent identification and the query coverage columns. Is the cleanest, perhaps, of the four submissions. So I wanted to let you know that I generated this consensus output for the species identifications. And my criteria were species with the highest percent ID, which of course makes sense, and then species with highest query coverage, usually greater than 95%, and then percent ID greater than 99%, most of them, or if they weren't over 99%, then species within 5.5% of each other. So that's what this is all about. There are still some samples I'm working on, and I'm going to be sending them in for analysis probably tomorrow, and then maybe by Friday the results will be in, but maybe also by Monday. So some highlights from the data. Well, one of the highlights that I didn't actually write down here is that I have samples in yellow and reddish colors. Um, those are actually species that have been identified by the International Union of Concerned something species of concern. I can't remember the red list. It's right here, and there are two categories of species. There are those that are yellow, which are vulnerable, and then there are those that are red that are endangered. And I find it really interesting that some of our species came out as yellow or red, which adds color to the storyline for your paper poster. Um, this is the actual identifications, and of the species that were identified by genetics and also by what the stores tell you, there are one, two, three, four species that were not identified correctly based on the genetics that we have for those species. Two of those species are actually listed as endangered. One of them is a chicken. Um, that's an interesting one, and I double checked the data, and it sure is a chicken, which is kind of hard to believe that tuna was labeled as a chicken, or identified as a chicken. Be interesting interesting to know where that one came from. Um, so whatever the number is here, there are actually 37 samples, but as I said here, several of these results were for bait fish, and I don't want the bait fish to be included in this analysis. The final poster. I mean, I understand why they had to be collected, and I'm happy to have identified them. But this is about supermarket fish identified correctly. So we want to make sure that we're not including fish that were bait fish. And there's also the ones that Talia collected that are the yellow perch and so forth. And I'm not sure. Maybe you could tell me, Talia, if the fish were collected by you or actually bought from a butcher or a store. Um, if they were purchased, then they can be included. If they are not to be included. So if you caught those fish, then we will exclude them from this analysis. like I said, I'm going to be getting more data, so there will be more data to be included in this final presentation. But for now, we have store-bought fish that, for the most part, were identified correctly, but there were several that were not. Um, Remarkably, down here, we have a bunch of fish that were identified as bacteria, and that I have no clue why, but we got more bacterial DNA than we actually got fish DNA. And perhaps at some point in the future, I'll actually study those, resample them, and see if we can get fish DNA. But it's possible, because these bacteria are actually known to be found in fish. It's possible um, that these fish had a lot of bacteria, and the DNA, I don't know. It doesn't make sense. But we have some bacteria, so we can't really include those. I think that's it for this. So I want you, one of the storylines is to take advantage of this information about endangered and threatened species. As I said, I drew the data from the red list. And one of the things I did after discovering that there are several species for which, there are several samples for which multiple species could be identified, I thought, well, just how similar are these species? And this is an illustration of just how similar those species are. Um, so there are samples 2 and 3, which are about a group of fish called ciscos. Our TDI, the bluefin herring, I think is what it's labeled at, which is also known as lake herring. There are 3 other species that could have been included in that. They could have been that fish. Um, there's one that is only 1 nucleotide off, and then the other 2 are probably another 2 nucleotides off from the species for which it was identified. And those 2 other species that are only 2 nucleotides off are both vulnerable species. So the genetic information suggests that while this species is likely to have been what was in the store, it's also very possible that there were 2 threatened species. One or two threatened species could have been in that store as well. And this same goes for this group of fish. I can't remember, they're called, I think they're called redfish or ocean fish. There's one endangered species that's only off by 2 nucleotides from the one that was identified, that is sold in the store. And then these are tuna. And tuna, there are 3 species that are either vulnerable or endangered. Yellowfin is the most common species, but as you can see here, these other 3 species are not that far away in their nucleotide sequences. 6 nucleotide differences, 2 nucleotide differences, 3 nucleotide differences from yellowfin. That's not a lot. It's a very small percentage. So it's possible here too that while yellowfin is likely to have been in the catch, these other species, they could have been as well. So the emphasis here, I think, is on not so much did the species get identified correctly, but just how closely related endangered species are to very common species, and how a misidentification of these closely related species could have consequences on the um, population status of endangered or threatened species. And that is all I want to say for now. So just to summarize, we have 2 files, Excel files. These are your results. This is the actual data. And reminder that I'm interested in fish that were store-bought, and you're going to generate some kind of output about the percent misidentified as a starter, but then I think you should also focus on the implications of misidentifications um, as it relates to conservation of threatened and or endangered species. Okay, that's it. Thanks.